Good day, this is Professor Will, CBMD PhD. Today's Monday, November 15th, 2021. It is 8:54 and 28 seconds, p.m. Eastern Standard Time. Today's research, nascent polypeptides. So let's hop to it here. Nascent polypeptides stabilize ribosomes. For uninterrupted translation. Date November 15th, 2021. Source Tokyo, Institute of Technology. Protein production translation is a complex process involving machinery, called ribosomes. How do cells counter? Ribosomal destabilization, leading to premature termination of translation. The research scientists have uncovered a novel role of nascent protein chains in stabilizing translating ribosomes. They suggest longer peptide sequences, spanning a ribosomal exit tunnel, and bulky amino acid residues in the tunnel entry help stabilize the ribosome by bridging its subunits, ensuring uninterrupted translation. Proteins are the key players that regulate cellular structure and function. DNA, which functions as a blueprint for protein synthesis, is first transcribed into a messenger RNA, mRNA, which is further read and translated into a polypeptide chain, a newborn protein, by macromolecular machines called ribosomes. And here the ribosome essentially functions as a tunnel through which the train of mRNA passes, and within which amino acids are assembled in a sequential manner, depending on the mRNA sequences, to form a polypeptide. And certain intrinsic sequences in the polypeptide can, however, trigger premature termination of translation, as protein synthesis is an essential cellular process, and this event can pose a great risk, resulting in protein dysfunction or synthesis of complete proteins. In nascent, newly synthesized polypeptides, this interrupting sequence, which is rich in negatively charged amino acid residues, is known as intrinsic ribosome destabilization, IRD sequence. And with such sequences scattered across the genome, how do cells avoid such a premature termination and ensure uninterrupted translation? The need for a tunnel structure is non-clear, given that the primary function of the ribosome is simply to polymerize amino acids into a polypeptide. The tunnel architecture, which spans 30 to 40 nascent polypeptides in length, may have evolved to balance the stabilization and obstacles of translation, elongation. The researchers began by analyzing the proteome-wide profile of the bacterial model system, Escherichia coli, and identified IRD sequences across various proteins. By constructing sequences of varying lengths preceding the IRD motifs, they were able to show that the peptide sequences that span the ribosomal tunnel can counteract destabilization by the IRD sequence in a length-dependent but sequence-independent manner. They further noted that longer sequences were associated with better IRD lowering efficiency. Next, they went on to investigate how properties of amino acid residues in the nascent peptide and their distribution across the proteome influence, IRD, using various amino acid substitutions preceding the IRD sequence. They found that residues with bulkier side chains were able to prevent IRD more effectively than did smaller ones. Further, they observed a bias in the sequence of amino acids across the proteome. Interestingly, open reading frames that code for proteins were enriched in bulkier amino acid residues towards the N-terminal regions that are translated first. The researchers speculate that these bulky residues occupy the entry of the ribosomal exit site, thereby stabilizing the translating machinery by bridging the small and large ribosomal subunits. Further on abrogating specific proteins in the ribosomal exit tunnel, they found an increase in IRD, suggesting that interactions between the nascent peptide and ribosomal proteins contribute to translation continuity. And together these findings indicate an intrinsic regulatory mechanism wherein the nascent peptide, in collaboration with the ribosomal tunnel, helps maintain ribosomal stability and continuity and translation elongation. The research findings highlight a positive feedback system wherein the ribosomal tunnel is occupied by its own product for uninterrupted translation, The researchers report on the role of nascent peptide chains within the ribosomal exit tunnel in ensuring efficient protein synthesis. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.